Tonight, we're going to have our second speaker is Eric Holland, MD, Ph.D., Doctor. And actually, we recruited him here from New York, and we were just discussing how nice it is to not be in the polar vortex, but to be building an ark because it's raining. So Dr. Holland is the Senior Vice President and Director of the Human Biology Division at the Fred Hutch, and that's one of the reasons we recruited him here, and we are incredibly fortunate to have him. He is the Director of the Solid Tumor Translational Research. Do you guys know what translational research means? No. Okay, so what that means is we're going to study something in the laboratory, and then we're going to take it and use it in the clinic. So don't study anything that we can't use in the clinic with the patient. That's what it means. So it's something that goes from the bench to the bedside. He is the director of the Nancy and Buster Alvord Brain Tumor Center, because that's his area of expertise, and also the Chap and Eve Alvord and Elias Alvord Chair Neuro-Oncology at UW Medicine. He's a professor of neurologic surgery, and UW School of Medicine. So they make a lot of like, it ain't brain surgery jokes. Yeah, he's like, it is brain surgery. All right. His research laboratory in the Human Biology Division is an interdisciplinary program. Do you guys know what interdisciplinary means? Yeah, so we bring in many people from different areas of expertise. Encourages collaboration among the faculty with a broad range of expertise, from molecular and cellular biology to genetics and then to clinical research. The program has a unique structure that fosters laboratory-based computational and clinical research that yields discoveries which can be rapidly translated into treatments for cancer patients. So that's pretty cool. Is that cancer right there? Okay, that's cancer, guys. His work on the solid tumor translational research effort is to enhance understanding of cancer biology and therapeutic responses and identify personalized treatments that improve patient outcomes. So one of the reasons that we asked him to come and talk to you tonight is to bring up this idea, what is personalized treatment? This is a new idea in cancer management, is for us to take a look at your tumor and see what are the things about that tumor that could make it susceptible to killing with various drugs. It's your tumor, so let's look and see what your tumor has that will make it susceptible. The way we used to do it is we said, in general, breast cancer behaves like this with these drugs. But now we're going to see what your particular tumor will do. He earned his medical degree at Stanford and his doctorate in molecular biology at University of Chicago. He's a member of the National Academy of Sciences and the prestigious Institute of Medicine. Prior to coming to Seattle, he was at Memorial Sloan Kettering, and then before that at the University of Texas at MD Anderson. And so without much more ado, I'm delighted to introduce you to Dr. Holland. Thank you.
So what's the question I get asked most commonly, right? And it's amazing. It's like everybody asks me the same question. My family asks me like once a week, are we there yet? Have we found a cure to cancer? When are we going to find a cure to cancer? Do you have a cure that you're not telling us about? So on and so forth. And the problem with all that, the construction of that, is that it is a binary thing. It's like you have a cure or you don't have a cure. If you're close to a cure, but the reality is that's not what it is. So the reality is that this is a slower evolution than we would like, but it is what it is. And it looks something like this. There are some cancers that do pretty well and have gotten better over the years. This is a graph from 1970s till maybe 5 years ago or so, and you're looking at a 10-year survival rate. And so some of these have done, not only do pretty well now, but have increased in their survival rate, quite markedly. Like, for example, testicular cancer is better than 95, certainly better than 90%. Melanoma has gotten much better. That's mostly because we actually find the tumor and take it out before it becomes metastatic. If it's metastatic, at least up until relatively recently, melanoma didn't have a very good outcome. But things are changing, and I'll talk a little bit about that. with melanoma. Breast cancer, as you probably know, has a really good outcome right now, generally speaking. Of course, this is back in 2007, and now I think the 5-year survival anyway is up over 90% at this point. Prostate cancer is, you can, it's interesting just to watch this. So this is this blue curve here, made a sharp angle here right about the time people began using anti-androgens, and so you can sort of see changes over time. But although there are some of these tumors that are clearly evolving and getting better, there's others that are clearly not doing so well, including pancreas, which we heard about this morning, or just recently. Now, another way to look at the data is to ask not, what is the survival rate for these things, but how much have we impacted the survival rate over the course of the last, say, several decades? And so you can see prostate, melanoma, breast and colon, a few of these things have really done well over the last few decades, but not all. And you can see down at the bottom, again, brain, which is a thing that I do, and I'll talk about this evening, on pancreas. Lung is an interesting one, and it illustrates, some subtleties in here. This graph here is the 5-year survival, not the 10-year survival I showed you earlier. And in order to have a 5-year survival difference, if you came up with a new drug today, you wouldn't know that it was actually giving you a 5-year survival difference until 5 years from now, right? And so all this is sort of looking in the retrospectoscope, essentially, as to whether you've really had an improvement. And when it comes to lung, I'm going to talk a little bit about this in the next few slides. Historically, lung has been a problem, and it's been a problem certainly if you ask questions about a survival rate of 5 or 10 years out. But let me give you some examples of things that are happening in lung right now, which are actually very exciting, and may eventually transpire into things that are long-term differences in long-term survival rates. So one of the observations that was made for lung, but it's also true for essentially all these other tumor types, is that there's one word for it, which is lung cancer, right? But in fact, it's many different tumors, and they have different mutations in them, and they behave different ways depending on the molecular characteristics of them. And this pie chart here illustrates the search for mutations or alterations in the DNA that could be drivers, the cause of the disease, which also might be targets for therapy. And so in these colored wedges here, blue here is a mutation called RAS, which is the most common, and red is something called EGF receptor, and less and less and less, and so on. Now, some of these have actual drugs that are already available that could be used in a tumor like this. Others, although we know it's a driver, we don't really necessarily have a drug yet, and then you have this big wedge of gray that doesn't have any known mutation. Obviously, something is going on, we just don't know what it is yet. Okay, so imagine you had a patient with in the red wedge here that has a mutation in EGF receptor. There are several drugs against EGF receptor, and so although there hasn't been a whole lot of differences in the clinical trials showing survival, there's been some for standard chemotherapy. If one, for the right set of patients, uses a drug against EGF receptor in those EGF receptor mutant patients, and I should point out here that this data came from Renata Martens, who's one of the lung oncologists. But this curve here illustrates an improvement in survival. So what this curve means here is that at the time of diagnosis, which is right here, 100% of the patients are alive, and as time goes on, patients pass away, and this doesn't look very good, obviously, because it's in months, not in years. But getting a statistically significant shift in that direction means that a drug, generally speaking, is working. And so it turns out that EGF receptor inhibitors do shift the survival of EGF receptor positive tumors, mutant tumors. Now, that's the good side. Many of you out there look at this and say, now, wait a minute, this is 1 year, this is 2 years. It's still not a great curve here, but we're getting some things happening. So what's happening is the tumor is responding, but it's coming up with ways of generating resistance, and so we have to understand this from a biologic standpoint. Now, another example in a similar vein was this little green wedge here, which has a mutation in something called ALK, which is about 4 or 5% of the total. And this was an interesting situation where people identified mutations in this gene, in this subset of patients, and there already was a drug against ALK, not developed for lung cancer necessarily, but it was already in the collection of drugs that the pharmaceutical industry had. And someone said, aha, there's mutations in ALK. Let's see if we can use them against this particular group of patients with lung cancer. And in fact, this is the data. So this is something called a waterfall plot, which means each one of these bars is a patient, and the height of the bar or the depth of the bar tells you how much the tumor either grew or shrank when it was given this drug. And so you can see in this whole collection of tumors here, a lot of them, essentially all of them, shrank initially with the ALK inhibitor. And then if you come over to the survival plot, you see an even better shift in survival with this particular drug called crizotinib. Okay, but again, let's be honest, this is 1 year, this is 2 years, and so you end up with resistance to some of these drugs. But that's not to, if I had a choice, obviously I would, if I had an ALK mutant tumor, I'd want the drug because it's clearly a positive result in the population as a whole. And so this kind of an idea of finding drugs, small molecules that block some of the mutations that are known to actually drive the tumor is something that people are doing across multiple tumor types right now. And lung is a really good example, but it's not the only example. Melanoma has a very nice story with a drug against a particular mutation called RAF, and amazing results are coming from that. And so these things are coming along. This is simply an illustration using lung for this particular illustration. Okay. So another sort of thing or strategy which is out there has to do with immunotherapy, and you've probably heard of that. This is also becoming a very hot topic. People have been working on immunotherapy for a long time, trying antibodies, trying vaccines, and sometimes these work well. But there's something which is very hot right now as a topic, and that has to do with treatments which regulate the body's ability to generally modify the overall immune response to things like cancer. And if you forget cancer for the moment, you certainly wouldn't want the body attacking all of your normal cells. And so there's a system that is called a checkpoint, which prevents your T cells from attacking you and all of you. And this is just an illustration of a T cell and a cell that's teaching the T cell which cells to kill. And there's a bunch of proteins that talk back and forth. And a lot of this is inhibitory, just to damp down the process. The initial drug that came out in this space was an antibody against this thing called CTLA-4, and that actually has been approved. And it has some efficacy in melanoma and potentially a few other things. The thing which is actually potentially more interesting now is something which is not yet approved, but these are antibodies to these two components here which talk back and forth between a T cell and an antigen-presenting cell called PD-1 and PD-L1. And these antibodies really seem to be having some very interesting effects on not only lung cancer, melanoma, but a few other things. And these are all trials, and so these are not approved drugs yet, but we have to imagine that this is where some of this cancer treatment space is going. So there are, in this disease that has almost no responses under normal circumstances, there are responses, and some of these are stabilization of disease for up in some cases as high as 534 days. So people are in many different cancer types are wondering whether this isn't going to be one of the main things that we do in cancer moving forward. But it's clear that it won't be working for every cancer type, and even in a cancer type that it works well in, it will not necessarily work for every person. It's going to be a subset of people, and guessing whether this particular person is going to respond well to something like this is going to be, is a guess as good as any at this point. But we'd love to figure out how to figure out who they are. Okay, so all that sort of preamble to what I'm going to talk about as far as the disease that I worry about. And this is, if you look at that list of of tumors, the thing near the bottom is a glioblastoma, which is a malignant brain tumor. It's a disease which, as I'll show you in a second, hasn't had a whole lot of movement as far as survival differences over the last several decades. This is an example of, on an MRI scan, this white area here, and a surgeon's job is to go in and take that out. Everyone after that, the radiation oncologist, the medical oncologist, they treat what happens afterwards. And I'll show you that there has been some serious improvement in, if you want to think of it, cancer care in the last several decades. And it mostly has to do with making that first part of the process, which is the surgery, safer. And if you look back, before 1950, brain surgery for malignant brain tumors had a really bad outcome. Many people didn't even survive right around the time of surgery, but that's really changed. The surgery is much safer now. People have been doing this kind of thing of opening skulls for a long time, going way back, Peru, so on. We do things differently now, obviously. We open the brain, we're very safe about things. Once you open it up, there's actually ways of seeing in to identify where the tumor is. This is an ultrasound. This is the probe that shows where the tumor is right underneath the surface, and we can use that to figure out where we're going. Obviously, you don't want to go in the wrong place, and this helps you get to the right place. Other technologies which help that is we actually have MRI scanners in the operating room now. And so we can operate on somebody, and if you're not sure you really got all the tumor out, you just get a scan while the patient's still asleep. And then if you left some behind, you can make sure you get it before they wake up. We could use MRI guidance in the operating room, which is a way of taking an MRI scan and reformatting it into a virtual three-dimensional image, which identifies where the tumor is in 3 space. So when you're operating, you actually know exactly where you are because of a recognition of where the tip of your instrument is relative to the MRI scan that's been reformatted virtually. We can identify where the tracks are. So the way the brain is structured is on the cortical surface is where a lot of the cells are that make us do what we do, but their fibers run down through the spinal cord. And if you cut those fiber tracks, then you have an injury that's very hard to recover from. But we can map those tracks out, and we can map them out in the operating room so we can avoid that. So this is an actual image that is in the operating room where this thing here is the tumor, and this stream of orange here is the fiber tracks that when we operate we have to stay away from. And as you might imagine, there's tumors near the language area. And so we can actually map out where the language is by doing an operation with the patient awake and talking to us, and we can figure out by stimulating certain areas that this is the part where you have naming errors, and this is the part where you have the ability to move your mouth and so on and so forth. So we stay away from all of that when we operate, and we actually will operate with holding the conversation with the patient at the time. And there's also, this is the last clinical brain tumor slide here. There's technologies that allow us to get an MRI scan while the patient is talking and map out on the MRI scan where the language area is. And this is an example of that. It's called a functional MRI scan. This area here is the tumor, and all these colored dots here are areas of the brain which light up and activate when someone is talking. And so you can see that one could, in theory, take this whole tumor out, and as long as you didn't touch these colored areas, they're not colored in the patient, but they're colored on the scan, right? So where you're operating, you say, I got to stay away from that. And in fact, this was one of my cases. That whole tumor came out. The patient was talking the whole time. He woke up. Everything was great. And the patient did really well. And so you'd say, well, we have evolved cancer therapy to be a real home run here. But the problem is, it's for a while. And the biology of this disease is what ultimately catches up to us at this point. And this is survival curves. And this is what happens if you don't do anything. So you know someone has a malignant brain tumor, and you don't treat them, essentially. And that's what it looks like. This is 1 year. If you do surgery really well, like I described, you get as much of the tumor out as you can, but you don't do anything after that. This blue curve is what the survival looks like. If you add radiation to that, it gets better yet, but it's still, obviously intersecting the axis down here within a few years. And if you add chemotherapy to the radiation, this is what it looks like in the black. Now the problem is that all the colored lines there were published back in 1967. And the black line was published in 2000 when I was at MD Anderson. And the study, the big study, which defines our standard of care currently, is this one in 2005. And you could see that basically what it said is that if you add temozolomide, which is the chemotherapy, to radiation, you get a marginal improvement in survival. So that is now the standard of care. And that is essentially the same thing as we had there, and not that different than everything else for the last 50 years. Okay, so why is that, right? We're doing great surgery, and yet we're up against a hard problem. So several things. Number one, it turns out that there's not just one glioblastoma. It turns out there are several. If you do molecular analysis of them, they fall into at least three major groups. Not every tumor within one of these groups is identical by any means, but they seem to cluster and behave in similar ways once they're in these groups. But they all kind of respond to, at least apparently, standard treatment kind of the same. And so it's, one might argue that these things may or may not matter so much. But something's mattering. There's a problem. We don't understand the biology well enough for this disease. So what would we really like to know? If you could just say, well, tell me what, wave a magic wand and I want data that would help me figure out what to do about this disease. So you'd like to know what's the cell that actually goes bad and ultimately gives rise to this tumor, referred to as the cell of origin. What are the mutations that actually cause the disease? Now, there's a lot of mutations and altered stuff in tumors that are not right, but they don't necessarily cause the disease. So the actual driver of the disease is probably a pretty good thing to know about when it comes to therapy. Maybe these subtypes matter as far as how they relate to each other. Maybe they don't. We don't really know necessarily right now when you give radiation, for example, to somebody, how many of the cells actually die? What type of cells don't die? Are they the cells that give rise to recurrence? Why do they behave the way that they do? There's a lot of biology questions that you really would like to know, and a lot of that is going to come from mouse modeling or complex biologic experiments that are designed to really address questions we need to know to decide what we're going to do clinically. So my laboratory, over the last, say, 10 or 15 years, has been trying to figure out ways of identifying the genes and the starting cells that are required in combination in a living animal, in a mammal, to generate essentially the same disease that we get, which is a glioblastoma. And so the way that we have done this is to take a human tumor, take the genes we think are probably the drivers, turn each one of those genes into a virus that we can then infect a mouse with, and the mouse is designed to be infectable by that particular virus. And most of the genes that are mutated or differentially expressed in this tumor don't actually do anything to a mouse, and they wouldn't do anything to you, but probably, but a few of them actually do. And when they cause tumors, those tumors look very much under the microscope like a human glioblastoma. All sorts of things about it. And one of those things one of the things that the mouse has really helps explain why a surgeon can't cure this disease. And so this is a mouse's brain. This circle here is around a very small tumor we identified that basically wasn't causing the mouse any problem. But this particular way that we've modeled, every tumor cell can be marked with an antibody, so we can see where they all are in the brain. And you, under the microscope, under this kind of a stain, you say, well, all the tumor cells are right here, and everything else looks basically fine. And if this was a human and I was a surgeon, I would go in there and I would cut this out and everything would be fine. Except for the fact that the tumor cells are actually all over the place. You only see them here, but they're all here, they're up here. All these brown spots are where an individual tumor cell is found. And so if you imagine translating this to people, cutting this out is never going to help, really, in the long run. It might help in the short run. People might feel better for a little while, but all of this is all over the place. And that's a problem that we need to figure out how to deal with when it comes to some of these diseases, and gliomas are definitely one of them. The mouse models can be used to understand, how therapy really works. And so here's a brain tumor in a person, here's a brain tumor circled here in a mouse. We can treat these mice with radiation just like we do people. The tumors shrink, but then they come back and they kill the mice just like the tumors, when they come back, have the same result in people. And this is, one person across time. This is one mouse across time. And here is the large tumor that eventually killed that mouse. And so if you now go to survival curves of mice, what you see is that untreated, a whole bunch of these mice eventually, in this particular way the experiment's done, they eventually pass away within a few weeks of being sick. And that chemotherapy makes that shift over, that's better, and radiation is better yet. And so we are in the process, and others are in the process of trying to understand how to make radiation and chemotherapy work better now that we have a system where we can really do those experiments. And where we can do other clever things, like we can engineer mice to with genes that come out of fireflies, for example, that make tumors glow in the dark. And that sounds neat and so on, but it's actually useful. So here's a tumor. This is the light coming out of this particular tumor. This light can be designed to tell us things like, is the tumor proliferating? Is there a particular signaling pathway that's on? And the amount of light, the number of photons coming out actually is a measure of that. But this is a live mouse, and the light is coming right out of the top of its head, and it can be measured. And this is the same mouse, and this is a radiation-sensitive tumor. After radiation therapy, that light disappears. The mouse eventually dies when the tumor recurs, but now we can actually start measuring things. And then we realize that the tumors are very complicated. They're not just a bunch of homogeneous cells mixed into the brain. They're actually really complicated. So this is maybe more details than you need, but here is a picture of the tumor. It has a structure that looks like this in the middle of the tumor, and has another structure that looks like this. The vessels, the blood vessels, are in this area. And in this area, there's many different cell types. There are things called stem cells, there's things called astrocytes and microglia, and a bunch of other things in that space. And this simply marks the fact that these are cells that are actually very resistant to therapy, which I'll show you in a second. And if you stain with a different antibody that stains sensitive cells, they're located in this area that's not around the blood vessels. So if you hit these, or if you treat these mice with radiation, these sensitive cells preferentially fade away within a few days, and these more resistant stem-like cells are enriched because they're not dying. And you can do measurements and so on and so forth, but we need to understand why these cells don't die with radiation, because if we could make that work better, then we could make radiation work better, and from what I've shown you, we could shift the survival curve maybe substantially if we could just make that radiation work better in glioma. Okay, and this is the last sort of mouse slide I'm going to show you, and that other things that we can do is we can use these mice to add to radiation to figure out something that either makes it work better, or maybe there are things that we're giving right now that make the radiation work not as good. And I'm not going to go into the details of this, but this is a particular drug that doesn't do anything to the mouse all on its own, really, for survival, but it blunts the efficacy of radiation substantially, and all of a sudden we need to start worrying about all the details of how we're treating people based on mouse work. Okay. So, translational research, what is that? Lots of things. So we can use mouse models to do the things that I just told you about, to really understand the biology of therapeutic response in cancer. We can analyze human tumors by molecular analysis to really understand all the complexities of the DNA, all the gene expression profile, and so on and so forth. We can do clinical trials that are based on these things to really come up with something that is maybe a little bit better, hopefully. Certainly, if you come up with a great idea, it's going to have to go through a clinical trial to find out whether it is better. The FDA is very important, but it sure does slow things down. And because it slows things down so much, the development of drugs are very expensive. And so the whole process is both long and slow and expensive. And it's a problem. On the scientific end, we can come up with ideas really quickly. We have to slow down when it comes to actually figuring out whether it works in people, and then if we can actually give it to people. So this whole process has been turning around and around, and it has gone through many phases where people came up with ideas that looked like they were the next best thing. It was the thing that was going to solve all the problems. Monoclonal antibodies went through this phase. All of a sudden, we thought, monoclonal antibodies, we got the right one, we're going to cure all cancer. And then small molecule inhibitors, as I showed you, work to some degree, but maybe not as much as we'd like them to. Other ways of modifying gene expression, something called siRNAs, which I won't talk about, but other types of immune therapy like T-cell things that I talked about. So all these things had sort of fads, I guess. Everyone thinks this is the answer. And it turns out that actually in some cases that is the answer for a very small subset of patients. And so this gets to the issue of what is personalized medicine. Is your tumor going to respond to one of these things? And how do you sort that out? So one version of personalized medicine is to be able to look for mutations across the entire genome of a particular tumor and find something that actually has a drug associated with it. And this is a very nice example from Colin Pritchard here at UW, where he's generated a chip, essentially, that will sequence several hundred genes. Some of them, as you can see on the top, actually have drugs associated with it. You have this mutation, there is already a drug available. Others are things that might have a drug in the near future. Others are things that might be important, may not be important. So it's several tiers of that thing. That is something which we can use to analyze anyone's tumor at this point. And in fact, it's taking off, and lung and colon cancer are the two that are most of the types of tumors that have been analyzed with this particular technology. But others, as you can see, are. And in many cases, there are genes found and that could be taken to clinical trial or actually simply given to patients if the drug's already available. Okay, so that's one version of personalized medicine. Find the mutation that has an associated drug. Okay, the second version of personalized medicine is this observation, and that is for essentially every clinical trial on drug X, whatever, you get this kind of a curve, and that is most of the patients don't respond very well, but a few of them do. And if you knew who these were, you'd be in pretty good shape. But because there aren't that many of them, then the overall trial fails because there's less than a 15% response rate. You say, forget it. Well, don't forget, there are some people that have really good responses, not infrequently. And so one of the efforts that are going on now across the country is to look for these rare responders and do deep molecular analysis to try to figure out what it is about them that would be a fingerprint for a good responder to the particular drug that was used. Okay. And so, and it may be that this would be only, 5-10% of the patients out there that you could say, well, this drug really works for your tumor, but this is sort of retrofitting. You don't really have to know what the mutation is that matters or why it matters. It's more empiric as opposed to rational. And the final version of personalized medicine, and all these answers are right. It's not like one versus the other. It's all ways of thinking about this, but it's big data. And big data is the buzzword these days. And the idea here is that there are lots of people with cancer, and there are lots of people whose full clinical record is digitized and available, and they could have their tumors molecularly characterized, and huge databases can be generated where is patient-centric, and each patient has their molecular data and their clinical data connected to it. And now we can ask the question, if the next patient comes and you analyze their tumor, what are the patients that have the most similar tumors to that one, to the one that you used, the new patient in your clinic, and how did they behave? How did they respond to therapies? All right, and I'm going to show you examples of that in a second, but this is the big data idea. Okay, so to do this, certainly nationally, but even within Seattle, it's a bit of an issue because there's so many databases, so many clinical databases spread across multiple institutions that have to be fused together. And so we are doing that here. And the biggest challenge right now is getting all this data into one place. And there is a system called Hydra, which is taking off right now. It's coming out of the Hutch currently, and it's run by a team of people. Paul Fern is the sort of thought leader here, but there's lots of people who do different parts of this, and they either identify clinical data and enter it into a database, or they train natural language processing systems that will extract data from the clinical database, or they pull out tumors and do molecular analysis on them, and so on and so forth. And this sounds fine. The structure looks something like this, where this is the University of Washington, this is the SCCA, and all of these boxes are different databases that have to be fused together and to create this thing in the middle called Hydra. And all these arrows are basically pipes that take all the data and fuse it in. And this is all run by Tony Bazzuti and his group of people in the bioinformatics core at the Hutch. And LabKey is the software designer. But the idea is that the system can actually read through the clinical data and find, because of the context in which a word is located, extract out, and with very high accuracy, probably better than a person actually, then recreate it into a searchable database like this. And there are pipelines of how that would actually occur. And if you think about it, there's more to this. There's actually issues of legal issues, and there's issues of privacy and so on. And so, all this has to be thought of when you create something like this. And so this is simply a diagram that illustrates how these databases are all living within a shell, a privacy shell, that the data is being pumped in from various sources, and there has to be legal agreements between everybody and memorandums of understanding of how this is going to work. And then the data leaving this population of databases has gatekeepers that have to be, and the people that would extract the data have to have the right permission to do so. The pilot here is something called Argos, but the people who are going to have access to this have to have password protected and treated for privacy and so on and so forth. But the idea is that you will have, and we already have, big databases where you can choose a particular organ site like brain, and it will pull up all this kind of information that you could, as a clinician, go in and try to figure out everything that's in that database so you can extract information to help you take care of this one patient you're interested in. This is going through some of these slides showing that the kinds of things that we're able to do. The interactive reports ultimately is a concept thing, and I wanted to talk about that finally at the end, and that is that if you imagine hundreds of patients, maybe thousands of patients, each patient's tumor being molecularly characterized and then displayed in some sort of array like I'm showing up here, each dot being a different tumor, and its location and how close it is to other tumors relative to how similar the molecular characteristic of that tumor is. And so this is simply a reference database. And if this is the new patient here that comes in, you say, well, how should I treat this patient? Should I treat this patient based on what we know about all of glioblastoma? Or can I treat this patient based on patients that are much more similar to that? And the overall survival for all of glioblastoma is this curve that I showed you earlier. But that's not what I want to know. I want to know the related things. I want to know everything in that circle. And of course, you could make that circle bigger or smaller depending on how many patients you need and how similar you need them to be. But then you can just simply get rid of everything else, and now you have a subset of tumors that are all similar to the one that you want. And now you can ask, for that subset, how are they doing? Some do well, some do badly. How did the ones that do well, how did we treat those? And let's treat this new patient like that, whatever that is, and it could be empiric, right? And these guys up here that did really badly, let's not do that, right? And so this is the other idea, the third idea of big data that is associated with personalized medicine. So this is my last slide, and this is simply imagining the future. right? And I don't know whether it's 5 years from now or 7 years from now or whatever, but it's going to look something, I suspect, like this. That we're going to know a lot more about cancer biology than we do now, about the biology of therapeutic response, about what we should be doing for individual cell types. That each patient, as they come in to be treated, has surgery, and that tumor is analyzed molecularly. And then if there are mutations in that population of that data, that would imply that a given existing tumor drug be used, then that's what will happen. Otherwise, that population, or in addition, that population will be compared to a bunch of similar tumor patients. And I think that would not just be in Seattle. I suspect it's going to be national. And figuring out how to make this a national database is going to be difficult, but it's really important because the numbers here, it's a numbers game. If we can get tens of thousands of people in this database, we can treat this like Amazon. All right? We can treat it in the same kind of analysis, big data analysis. And if you don't have enough patients, it's a little bit difficult. So we're trying to figure out how we're going to actually make that happen. And finally, the mutations are in tumors, obviously, but there's actually data out there suggesting that we might be able to get molecular data right out of the blood. There may be enough DNA circulating in the blood that we can sequence it and know what's going on in the tumor. So we might be able to follow people over time. Are they responding appropriately? If they're not responding appropriately, switch to something else. When the tumor recurs, what's its characteristics, and make decisions about that. And that would be great if it could come out of the serum. And my guess is, rumors I hear say that that's actually what may happen coming up. Okay. That's my last slide. I thank you very much. I was thinking of putting the jokes at the end, but I put them in the beginning just to make you like me first. Thank you.
You know what, I actually think today's topic, this big word of cancer, was incredibly uplifting. I think it was uplifting the first half in terms of the progress that we make in communicating to our families and our patients with a cancer diagnosis. And this is absolutely amazing, just absolutely amazing. I deal with prenatal diagnosis, as you know. We are in a stage where we can isolate, fetal cells or fetal DNA in the maternal serum and provide prenatal diagnosis on the fragments of fetal DNA that's floating in a mom's serum during her pregnancy. And this is really another example of all the technology. And I feel like this is incredibly uplifting in that sense. So questions?
How do you get the word out to rural areas about all these improvements in cancer treatments?
So the question is, how do you get the word out? It's not so easy on the one hand. On the other hand, it was in the previous talk, it was brought up that the internet is really powerful, and I think ultimately that's the thing that will normalize everything, and that's just we have websites associated with the work that we're doing, and the real question is, how do you point people to those websites? That's probably a better question, because that's ultimately going to be the answer. Yes?
You mentioned a national database. Maybe I missed it, but what kind of timetable are you thinking of for that? What do you expect?
Yeah. So right now, I can tell you there's a consortium of the main cancer centers, which is the Farber, Anderson, Sloan Kettering, and us here, to try to figure out how to, initially with those centers, figure out how to do this. And this is probably going to take maybe a few years. But I think what's going to happen is, we'll build it and they come. So people are going to almost have to do this because it's going to become the standard pretty quickly. It just isn't there yet, and maybe I'm over and optimistic. But I think that if we can build it right, actually Seattle's a perfect example because we have to figure out how to get multiple institutions to work. And if we can do it here, that's really the beta test for. It's harder for us than for Anderson, for example. It's one entity, so that's.
But you don't have any guesstimate as to roughly how long it might take?
Well,
I mean,
It
I
depends on
love
what you mean
this
by it. It's almost like, my first slide, are we there yet? So, I mean, by this summer, we're going to have a pilot. We're going to have this pilot by the summer, all right? But if you really want to know when it's going to be usable, really usable clinically, it's going to be continuously better and better and better as it gets built out. So when it's really good, I think it's going to be a few years from now. But this summer, we will have the pilot working.
Okay. I love the idea.
Okay, thank you. Yes?
So in your imagination of the future of every patient's tumor being analyzed,
Yes.
molecular, where do those patients stand today? Who are those patients? Are they just patients who come to the University of Washington? Are they in clinical trials?
Right, that's
And
a great question.
Who pays
and
for
who
that?
pays? That's really the question.
Thank you.
So let me deal with that first, because I think that actually will drive everything. So the price on all of this is falling rapidly and has continued to fall. If you look backwards, I don't know what the rate is, but it's shocking how quickly it falls. But it is reasonable to assume that within 12 months, certainly 24 months, the cost of a full analysis will be less than what we're currently paying for the standard tests that we're doing. You can sequence the entire genome at this point for a price that is comparable to the cost of doing 1 or 2 gene tests. It's just different technology, and it's getting better and better. And if you fast forward all of it is going to be a few $1,000s. And so the hope then is that the insurance companies are going to say, well, we could pay several $1,000s for 2 genes, or we could spend less than that for everything. So we hope that this will result in them paying. And once that happens, it's just going to be what happens. I mean, it's going to be like any pathology readout. That's going to have everything in it. But that is a leap of faith that the insurance companies are going to realize it's in everyone's interest to do this. I think they will.
You mentioned enhancing things with radiation quite a few times, but you didn't mention what kind of radiation. And I'm wondering whether proton beam therapy has a role here, or ion beam therapy,
Sure.
and these things. It seems to me that for small tumors, like in the brain, that's the ideal place.
Sure. So, I mean, just so everybody knows, there are types of radiation which will radiate the entire brain, and that's one extreme, all the way down to some incredibly focused technologies like proton beam and so on, that have very little spillover from where you want it to radiate. And depending on where the tumor is, and what is next to it, and the stuff you want to actually preserve, well, that's really the real issue. You might have to go to things like proton beam that are very, very precise. And in the brain, as it turns out, for glioma, you actually don't want to be that precise because of all the scattered tumor cells that I showed you. You kind of want to hit a general area when it comes. So different tumors are different, but there's clearly a role for proton and other sort of beams. And the truth is, we don't really know all the various things we could do with each one of those, and all that's an evolving process. But that
MD Anderson has a lot of experience with that. That's why I was wondering.
sure, but we have a lot of it here too. We got a proton facility here.
Brand new on proton beams here, but
Right.
MD
I mean,
Anderson-
the proton facilities are, they're all relatively new. I mean, they're in the last decade or something, and so it's all evolving rapidly. Yes.
Hi, thank you. I think this DNA serum sequencing is rather interesting, and I'm wondering if it's going to have an application in screening, like if you can just go to your doctor and
Maybe.
have a vial of blood
Yeah.
taken and...
Yeah, I mean, so the issue with screening, the issue, if you compare screening to taking a patient that you know has cancer and you want to follow something, if the patient you know has cancer, then the signal to noise is a little bit different than if you look at a population of 100,000 people to identify a few people that have a problem. If the noise is high enough, you pick up a whole lot of people who don't have the problem, right? And so, for a population of people that are at high risk, something like this may be really good. But the truth is, we don't know how much noise is in the system. It could be that this sequencing techniques are actually going to be very low noise, and it could be used for sequencing. We just don't know.
So maybe like in combination with a mammogram that has a...
Maybe,
something like that?
I mean, certainly if you saw something, on a mammogram and say, oh my gosh, what's that? There might be, at that point, it's a high-risk population. You might, might be worth it. Yeah.
Thank you.
You talked about the database and all of our sort of cancer clinics in this country. Do you reach out to the rest of the world? How is that
Absolutely.
working?
Absolutely. I mean,
You know, we're just-
yeah, you know, we're definitely envisioning that. Now, the platforms to analyze the tumor tissue is the same across the planet right now. What's not quite the same is the clinical care and the quality of the clinical data. But we're in real conversations with people in India and to some degree in China, because you know any place that has a huge population would be really helpful for the big data thing, for sure.